Good afternoon, ladies and gentlemen. Welcome to Carestream Molecular Imaging's presentation introducing the in vivo Xtreme. Due to the number of people here today, we have muted all attendees. If you have any questions during the program, please type them into the question box to your right. Where it says Send To, choose Send Questions to Staff. At the end of the presentation, there will be a brief Q&A session. We'll try to answer your questions, as many as possible during this time, and we'll contact you directly if we're unable to answer your questions online. I am now pleased to introduce you to today's speaker, Dr. Seth Gammon, Global Product Manager for In Vivo Imaging Systems here at Carestream Molecular Imaging. Seth?
Hi, good afternoon, everyone, or good morning, or good evening, depending on where you're at in the world. My name is Seth Gammon, and I'm the Global Product Manager for Carestream Molecular Imaging for our In Vivo product line. And today I want to spend some time speaking with you about our new In Vivo Xtreme. So very briefly, I want to introduce who Carestream Molecular Imaging is. Carestream Molecular Imaging is part of a global company called Carestream Health, or Carestream. It's an independent company of about $2.5 billion in revenue. We are a strong global company that not only employs 7,200 people worldwide, but we also do a lot of innovative research and development, as you can see, from over 1,000 patents in technology and IP. Carestream Health has a lot of different divisions. One of them, us, is molecular imaging. There's also a medical imaging group, dental and dental software group. There's a very large RIS and PACS, or Healthcare Information Solutions group, that delivers solutions to hospitals worldwide. We also have printers, especially for printing medical films. And if you've used Kodak scientific film, then you've used the Carestream product. We're located globally, both from a manufacturing and a research and development perspective, as well as from a service perspective. So if there were anything wrong with your system, we can get to your site and do repairs on site. Beyond that, we also have a multi-tiered service organization that allows us to help you triage problems with remote access technical support, telephone support, as well as PhD level application scientist support to help you tune up and optimize your systems or your new experiments. But today, I wanted to spend really all of my time talking about the new In Vivo Xtreme from Carestream Molecular Imaging. Carestream Molecular Imaging was the first company to bring an all-in-one imaging system to the market with high-resolution optical X-ray combined with radioisotopic imaging in just one system. Recently, we also introduced the Albira, an innovative combination of PET, SPECT, and CT to the preclinical market space, and we continue to invest in our research and development dollars into this space to bring new products and accessories to market. And as a recognition of this, in fact, in 2011, Frost & Sullivan, an independent third-party analyst group, recognized Carestream Molecular Imaging with the 2011 Product Line Strategy of the Year Award. And this was for our MSFX Pro, F Pro, FX Pro, and DXS Pro combined with Albira that provides 7 preclinical imaging modalities in 2 systems to our customers. What they didn't know at the time was that we weren't done innovating. And in fact, we just recently introduced the new In Vivo Xtreme. The In Vivo Xtreme combines fluorescence, luminescence, radioisotopic, and X-ray, along with a unique choice of cameras, both a front-illuminated camera and a back-illuminated camera, so that you can match your research needs to your budget, get the system that you need now, and if you want to, upgrade later, for example, from front-illuminated to back-illuminated, or if you don't need X-ray right away, you can purchase a system without X-ray and upgrade all in the same footprint to X-ray later, all done in your lab without changing the footprint of the system. So now when you look at the lineup of imaging products from Carestream Molecular Imaging, you can really think about it in two parts of the portfolio. One is a classic benchtop model. That's our In Vivo MSFX Pro. That contains a front-illuminated 4-megapixel camera and now is available with a fixed lens option. Or you can go to our tower-based new Xtreme system that's available with a choice of cameras, really depending on your need for luminescence sensitivity, and your need for a very high resolution X-ray. And those can be matched to the budget that you've got and again upgraded later in the field. This is the Albira system is a perfect complement to any of our optical and X-ray systems. If you need truly quantitative 3D tomography, you can configure the Albira in any combination of PET, SPECT, and CT that you might need and upgrade later in the field if you needed more imaging modalities. So for example, I might only need to start with a PET CT because I don't have SPECT probes ready to image in my animals, but I know that my research might go there a few years down the road. The system can be configured as a PET CT, and when you're ready to upgrade, the SPECT cameras are upgraded at your site. The system is normalized and ready to go. So with either the MSFX Pro or the Xtreme, you get 7 imaging modalities in just 2 systems. That means there's really no limit to the different combinations and permutations of experiments that you might run on your small animal models. And no matter what new probes become available in the emerging space of molecular imaging, you'll be ready with the imaging modalities at your fingertips when you need them, when those probes are available. So now I'd like to introduce the in vivo Xtreme. First, we'll start out with the disclaimer. There's a lot of different intellectual property that both ourselves and others hold in this space. And that's just something you need to be aware of and keep in mind during your purchase process. So what is the Xtreme? Then we'll go into a few imaging applications, and then finally introduce the concepts of spectral and mixing in Mars, which are a standard part of all of our Xtreme systems. So let's take a quick tour of the inside of the Xtreme. And we'll start at the top and work our way to the bottom. The system comes with a true microfocus X-ray head. It's the only optical and X-ray system out there that has a true microfocus X-ray head along with our MSFX Pro. It's got a very high industry-leading for multimodal imaging 500 microamp X-ray head that allows you to collect images for CoRegistration in just a second. If you're used to looking at human X-rays, this 45 kVp number might seem strange, but that low energy X-ray is designed for getting nice bone and soft tissue contrast in mice and rats as opposed to human bones where you need much higher energy. The system also comes standard with the geometric magnification stage that allows you to very rapidly take extra high resolution images by bringing the sample closer to the X-ray head. Would be located up here, and that would then not be able to be co-registered with fluorescence and X-ray. If you acquire X-ray images here at the platen, then you can co-register any of the different modalities with X-ray. Moving down, we have the animal management zone. This is where, whenever you're acquiring your image, the animals are housed. As you see in this image, we've got our patented industry-leading ultra-thin, ultra-uniform radiographic phosphor screen that can automatically slide under or out of the way of the mouse. And that's one of the things that enables our system to acquire very high-resolution X-rays. If you're interested in high-throughput screening of PET or SPECT probes, we also provide a high-sensitivity radioisotopic screen. That screen is thicker than the X-ray screen, but can convert more high-energy gammas into visible light to be captured by the camera. So you could put that in there, and then it would also automatically slide underneath the animals when you move into radioisotopic mode. One of the differences between this system and anything else that's really out there is that we use a bottom-up imaging geometry, and that's important for a couple of reasons. One is that if I have mice located next to each other in an image, multiple mice, my excitation light for my fluorescence all comes from the bottom on a nice flat surface because gravity flattens mice out. That means that I don't get shadowing of excitation light from one mouse to another that you can have with top-down imaging systems. In addition, because this system, as you'll see in the next slide, uses very fast optics, you want to have a very flat focal plane to get nice high-quality images using those low F numbers. And by imaging from the bottom, again, mice, gravity just nicely flattens out the surface that you're trying to image onto the animal management screen. There are also light-tight ports on the side of the instruments for delivery of isoflurane anesthesia, as well as ports that allow you to do catheter injections so you can acquire time-lapse images of probe distribution or reporter activity. The system is also compatible with our multimodal animal rotation system, which I'll illustrate later, and of course has a platen that allows you to image up to 5 mice at a time quite easily. Down below the animal management zone is one of the hearts of the system, and that is the camera and emission filters. Below the system, you've got a choice of 2 cameras, both large sensor sizes to increase your luminescence sensitivity. One is a high sensitivity back-illuminated 4 megapixel camera. The other is a front-illuminated 16 megapixel camera when resolution more than sensitivity is one of your driving factors. A question that often comes up is, what is the difference between a front-illuminated camera and a back-illuminated camera? A back-illuminated camera is more sensitive because the circuits and electronics that read out the data from the sensor are not in the way of the light path. They're in fact they're located on the opposite side of the light path from the light coming from your sample down to the sensor. In a front-illuminated system, those wires and electronics are in the light path, and that decreases your quantum efficiency. Unfortunately, back-illuminated sensors are much more expensive to manufacture, and so there's a trade-off in budget and luminescence sensitivity. The good thing is that really unlike anyone else out there, we provide you the choice of cameras that meets your luminescence sensitivity requirements and your budget, and you can rest assured that you can upgrade later from the front illuminated to the back illuminated when budget becomes available and the need for extra sensitivity is there. Below that, below the stage, you have our 6 patented wide-angle emission filters that come standard. That allows you to image emission wavelengths from 535 out to 830, and there are other optional filters available, such as an 850 long pass filter, and those can be configured at time of purchase. Located below the camera is our really industry-leading broad spectrum light source, the 400-watt xenon illuminator. It comes standard with 28 excitation filters that cover the range from 410 to 760, so you can excite any fluorophore from the visible to the near-infrared. Now, while we recommend strongly that you start as far into the near-infrared as possible, we do provide these excitation filters so that you can image red and green fluorophores if you have to, while you're migrating to the near-infrared. We also provide standard spectral deconvolution software to help separate autofluorescence from signal when you're working in those green and red regimes. The in vivo Xtreme comes with 2 interfaces, a standard interface and an Xtreme, and an advanced interface. With the standard interface, in one single click, you can execute complex protocols that you've developed at your site. You can also, with a single click, capture multimodal acquisitions. So this screen is set up right now where you would select your modality, in this case fluorescence. A background or anatomical reference image comes up, in this case X-ray. You select your exposure time for the fluorescence image, your f-stop, your excitation and emission filters, and you click capture. And that's it. You're done. Then it'll acquire a fluorescence image, it'll acquire an X-ray image, and then overlay the 2 and co-register them for you. So if you acquire an X-ray and a fluorescence or a luminescence image, you can get a nice anatomical map because the fluorescence or luminescence or radioisotopic image is automatically co-registered with your fluorescence image. The Carestream molecular imaging software also comes standard with quantitative region of interest tools. So you can look at your functional image, you can draw a region of interest and quantify the data in counts, photons per second per millimeter squared, or picowatts per millimeter squared. In addition, we know that a lot of in vivo imaging, even after you have those nice quantitative units, really ends up being a ratio of an experimental to a control. And so with a single click, you can reference any region of interest in your image to another control region of interest, and all the data is normalized to that control area for you. Next, let's go into some imaging applications. First, we always think about X-ray, or talk a lot about X-ray, as a modality for co-registering anatomical and structural images, with our functional, fluorescence, and luminescence, and radioisotopic images. However, there's a lot of good science that you can do by using X-ray to study bone and soft tissue phenotypes. And here's an example of, in just 4 seconds, acquiring a very high-resolution image of a mouse paw on our geometric magnification stage. You can make out all the fine bones in the mouse paw. You can see individual hairs in the image, as well as the trabecular structure at the end of the bones. In 2 seconds, you can get a very detailed image of a skull that would allow you to look at things like developmental abnormalities or traumatic brain injuries as they affect bone, especially bone structure in the brain. But now let's move on from our strictly anatomical imaging to co-registering, I think very excitingly, not just one functional image, but multiple functional images with X-ray for an anatomical reference. In this case, we're using a brain cryolesion model where the brain, a cold block, metal block, is applied to the skull of a mouse, inducing an injury to the brain. After an hour, we inject PSVU 794, which targets dead tissue through binding anionic membrane, and that's read out using near-infrared fluorescence. We can also use our luminescence imaging modality to look at myeloperoxidase activity in vivo, so we inject 5 mg of luminol IP, and then we acquire luminescence, fluorescence, and X-ray, and then co-register all of that data. As you can see, there's a lot of luminescence induced both at the target site as well as some bulk inflammation going on in that whole mouse, probably due to mice barbering each other in the cage. And you can see very nice uptake in the right of the near-infrared fluorescent probe at the injury site. And you can co-register any and all of those modalities together quite easily. Of course, because this is non-invasive imaging, we can also look at these studies in a longitudinal fashion. This is 20 hours post-industry. Now that global inflammation has really dropped off, but you still see some, though decreased, inflammation at the injury site. But looking at cell death, there's still a significant amount of cell death from the fluorescence image. And of course, that can all be co-registered in real time. Another area that people are interested in looking at is Cherenkov imaging and the ability to look at different structures with light that's emitted by positron or high-energy beta emitters. One thing to keep in mind is that these Cherenkov images, and in this case a movie, is heavily weighted to the surface. And that's because Cherenkov light primarily is in the UV, some in the blue, and then tails off as you get to the red and the near-infrared. So with a high sensitivity luminescence system, you can use this property to look at PET and high energy beta emitters at structures that are relatively close to the surface. In this case, you can clearly see most of the activity is being cleared by the bladder, and you can see it in the heart. As an example of how this still doesn't, with Cherenkov, with any system, give you the full story, is that because we've placed the animal ventral side to the camera, we don't see the uptake of the FDG to the brain because the path distance for the blue light is just too far to get out. So while this technique provides an interesting pre-screening tool for superficial structures with PET probes, it's really nice to have the radioisotopic screen that we provide, and only Carestream can provide, so that you can screen not only PET probes and beta emitters, but also SPECT probes, which are a big part of the of nuclear medicine's arsenal of imaging probes. Another example of using high sensitivity luminescence is to look at myeloperoxidase activity as it's induced by a nutraceutical that was injected IIP. Now in this case you can see not only is there the injection site, but regional lymph nodes are also being have excited myeloperoxidase activity. This was co-registered not with X-ray, but with a reflectance or photographic image, which is also easily doable using the acquisition software in a single click. Because Carestream provides tools to export images really quite readily through batch export modules, you can also build RGB images that are co-registered with luminescence or fluorescence through the use of ImageJ. And here you can see regional myeloperoxidase activity that's been activated after a wound, as well as co-register that with a site of near-infrared fluorescence. We've talked a lot about the high sensitivity luminescence, but you can also get really nice near IR fluorescence images and really fluorescence images from the extreme. So here we've injected ICG, 1 nanomole of ICG into the tail, not a tail vein, but into the tail. And this allows us to look at where does it drain to. So it's actually highlighting if the pathway that this ICG can take through the body, where it's being cleared through the GI, but it's also able to find and see the local regional lymph nodes. And in fact here, with our nice high-resolution system and that bottom-up imaging geometry, you can see the fine structure of regional lymph vessels as well as the lymph nodes. Of course, you can look at activatable probes, which are really one of the most exciting near-infrared fluorescent probes that are out there, because now you're not just looking at a target to non-target that's dictated by the binding of a probe to a site and then clearance away from it, but rather an amplification of the signal at the enzyme site you're interested in by the enzyme itself. Now, last but certainly not least, I want to talk a little bit about how spectral and mixing can still help you with this system, as well as our multimodal animal rotation system. With that 28 excitation filters, you can acquire a data cube of as many or as few images as you might like with fluorescence imaging. Once you've got that data stack generated, the system can look down on a pixel-wise basis and see the observed excitation profile at that pixel. And then based on saved reference spectra, it can now separate that stack of data into pure fluorescence coming from our autofluorescence, in this case yellow, gut autofluorescence green, or 2 near-infrared fluorophores in red or blue. That can still be co-registered with X-ray because the mouse has never moved. All we did was take multiple different excitation filters and acquire images and post-process the data using software. That can also be, of course, co-registered with reflectance images as well. As proof of concept in an old multispectral, it's been peer-reviewed, published that you can use this to separate out gut autofluorescence from the probe that you're interested in looking at. Now, Mars is multimodal animal rotation. On the left, we look at a subject that was imaged in near-infrared fluorescence using OsteoSense 750, a bone sensing probe, and image with Mars. In the middle and the right, you see PET and SPECT images of different bone probes. Now, if you're using Mars, your eye really almost gives you a three-dimensional representation of the distribution of that probe. But only with quantitative PET-CT and SPECT-CT can you really get true volumetric measurements of the process you're trying to look at. One of the really great properties of Mars is that unlike traditional systems where you only get a single view of the animal, or quote 3D tomography systems where you only get a single view of the animal, you actually get to survey all the way around. So if you have a subtle signal that doesn't transmit all the way through the body, you'll miss it with a quote 3D system that only takes a single view. Whereas with a Mars system, as you can see on the left, as you cycle through to the optimal angle, you'll actually see your signal, even though it doesn't penetrate all the way through the body of that animal. So in conclusion, the Xtreme provides a powerful, very powerful, multimodal, high-sensitivity system. It has fluorescence, high-sensitivity luminescence, as high-sensitivity radioisotopic, that can be combined with very high resolution X-ray. With just a single click, you can acquire fluorescence X-ray images, luminescence X-ray images, fluorescence and reflectance images, and with our protocols, you can acquire any combination of any modality that you might like. That gives you really unprecedented flexibility for a high sensitivity luminescence imaging system to get the data that you need and know that you've future-proofed your labs, because as new probes come on the market, even if they're fluorescence, even if they're new luminescent probes, if they're new fluorescent reporters or new luminescent reporters or radioisotopic reporters, or if they're contrast agents for X-ray or CT, you'll have the tools in your lab to image those probes right away. You won't have to go out and buy another system. The Xtreme architecture is also the first and really only in vivo system that allows you to choose your camera at time of purchase and upgrade later. Finally, the Xtreme system comes standard with excitation-based spectral and mixing, so you can look at multiple overlapping fluorophores in the near IR, or if you have to image in the green or red, use that spectral deconvolution to remove the autofluorescence from the image. A really great optional accessory for the Xtreme is Mars, that allows you to have really a true 360-degree sampling of the data that comes out of your mouse. That gives you improved signal to noise and make sure that you don't miss a critical signal just because you weren't looking at your mouse from the correct angle. So I'd like to thank everyone very much for their attention, and I'll be glad to take any questions that you might have.